Can you start by telling us your name, your institution, and your role, please?
Yeah, my name is David Spencer. I'm at Washington University, and I am a PI on a UO1 project for the SCGE.
Great. And then can you describe your SCG research in a couple sentences?
Yeah, we're developing a genome sequencing assay to evaluate genome-edited human cellular therapies to ensure that the genome editing is performed as expected and does not result in any unwanted mutations that could be dangerous for a patient.
And then what specifically do you work on within your research project?
I'm the PI, so I kind of run the show. Work with postdocs and bioinformaticians who are doing the work. I also write software code myself and analyze data and interpret it along with the other PIs and co-investigators of this project.
What's your favorite thing about either this research or your research in general?
I think that the genome holds so much fascinating information, and this project leverages that to make sure that cellular therapies are safe and effective. And so, using the technology to its fullest to further genome-edited cellular therapies, I think, is really fascinating and exciting.
Great. And that's kind of a good segue. What's something cool or interesting or maybe unexpected that you found in your research?
Um, so I mean, I think there are often many questions that scientists and doctors might have about a cellular therapy, and the fact that we're sequencing the entire genome allows us to answer many of those questions in a very short period of time, and with very precise information. So I think that's really, really cool. One thing that's interesting and not necessarily cool is the range of unexpected mutations that can happen when a genome is edited with genome editing technology. And so, I mean, I think that it's interesting to identify these unwanted mutations and characterize them so that we know how to identify them in therapies that are going to be used for patients.
And then what's an advancement that you think will happen in genome editing or gene therapy in the next 10 years or so?
In the next 10 years, I think it's just going to become more, genome editing will become more and more common, a more and more common way that patients are treated for a range of diseases. I think it may be hard to predict which diseases will be the most effectively treated with cellular therapies, but I think it'll just become more common as the technologies become better, more efficient, and especially in our experience with the technologies grows, and we become, more comfortable with the technologies and are able to use them more effectively. So I don't know. I think it's going to be more common.
And then lastly, is there anything else that you would like to say about your project or the SCG consortium more broadly?
One thing that I think is really great about the SCGE is bringing together scientists and doctors and practitioners of this technology so that standards can be discussed and decided upon, so that going forward, everybody kind of has a common language and perhaps common platforms, so that we can seek to have kind of just a common sort of language for this technology, so that we can all communicate effectively with each other. I think that's a great benefit of this consortium.